Top Med Talk.
Hey, the residents are here.
Thank you very much. Welcome back after your break. We just had that fantastic talk from Dr. Molyneux, and we're going to bring it a bit further into the perioperative field now. We're just going to try and take some of the things or thoughts that maybe have been generated from what we've just heard and move them more into maybe what we're more familiar with as perioptimists and perioperative physicians. So we have a international panel of high caliber here, and Professor Grocott. So Professor Grocott, sorry, that was mean, is...
and a comedian.
Yeah. Thank you. Where's the cool question got now? Okay. So Professor Grocott, as many regular Top Med Talk and EPOM and Dukeites know, is a professor of critical care and anaesthesia in Southampton. He also has multiple other roles to do with National Institute of Health Research, etc., etc. His area of interest in preoperative exercise, testing, capacity, patient capacity, etc. Anything else he wants to tell you, he will. He's online. Next to him, we have Dr. Brant Inman, who is one of our distinguished Duke urological surgeons. He has a very strong background in cancer biology, particularly in urological cancers, and I think it's fair to say latterly has become increasingly interested in why his patients become sick after surgery beyond the cancer and the surgery itself. Next to Dr. Inman, we have Dr. Pastover, Amy Pastover, who is, again, multi-talented. She is a doctor of physical therapy, a PhD, immunology, etc., strong interest in rehabilitation and frailty, particularly in intensive care patients and in older patients. We also have Dr. Wishmeyer, Paul Wishmeyer, another regular on Top Med Talk, who is a professor of medicine and surgery at Duke, also head of perioperative research at the Duke Clinical Research Institute, and has extremely strong interests in preoperative nutrition. Rounding out the panel, we have Dr. Molyneux, who we've all just heard speak, and his credentials have already spoken for himself. So, without further ado, that's the longest introduction I've ever given. Let's get on to some questions. So really, we'll start with Dr. Molyneux. You talked very eloquently about the first step you learnt in gerontology, which was not the first thing I learnt in anaesthesia. It was how I learnt about the anaesthesia machine, actually. But this observation that two patients of the same age with the same listed comorbidities seem to have different responses to an insult, be it injury or a procedure. So just bringing that observation to the perioperative sphere, are you able to comment at all on how you feel mitochondrial biology might impact the observations that we've all made in our perioperative patients?
Yeah, I think the relationship of physical function to surgical outcomes works in a very similar way to why our bioenergetic profiling works. It's because, for example, if you think about a measure like gait speed, this integrates a lot of things. It's not just muscle function, it's skeletal muscle function, cognitive function, and all of these things coming together and reporting on, more globally what's going on systemically. And again, I think, the bioenergetic capacity measurements work the same way. The difference is that as a blood test, this can be done in a very precise manner. It can be done in everyone. We can't ask everyone to stand up and walk. We can't ask everyone to do an SPPB, but in most patients, we can take a simple blood test.
Right. And I can see one of the slided questions here being, how close is this to being a real preoperative lab test?
There's a lot of work to be done. I think, to be honest, we're still in the early stages of this. One of the things that we're still trying to figure out is to really define what a healthy bioenergetic profile looks like. Right now, the tests that we've been reporting on, we've been focusing in on just a mixture of peripheral blood mononuclear cells, and globally what they're doing with regard to the respiratory capacity. In our newest iterations of this test and what the NIH has been giving us support to do, we're actually breaking down the individual blood cell populations, looking at the respiratory profiles of monocytes, lymphocytes, platelets, and then relying on machine learning algorithms to really identify bioenergetic signatures that predict the outcomes that we're interested in. And those could be, a whole host of different outcomes.
So, Dr. Inman, you're a surgeon on the panel. You are aware of how frailty impacts your patients' outcomes. What are your thoughts on what we've heard so far this evening?
Well, I think it's interesting. I have a few questions about it. Most of the data that was shown pertains to the ability of mitochondrial metabolism to approximate some better metric, like a 6-minute walk test. Or a 6-minute walk test is cheap. A seahorse test is not, probably. Do you think that your metabolic profiling is going to be better than existing frailty measures that you can do by simple questionnaire or get up and sit and test and stuff like that, or do you think it's going to be just the same?
So maybe the best way to answer that is to tell you how we pitched this to the NIA. We pitched this as, in 5 years, our goal is to come up with a $25 test. And by identifying more specifically, like an individual bioenergetic profile that we need to measure, rather than kind of globally looking at mitochondrial function, and this is something that can be done with more affordable respiratory profiling techniques as opposed to the seahorse that you mentioned.
So, Mike Grocott, we have heard you speak many times on Top Med Talk and at TRIPOM. Your specific interest is in preoperative exercise testing beyond the 6-minute walk test, and this is something I know we've been discussing with Dr. Inman at Duke. Are you able to shed any more light on maybe how a CPET test might add more than even a 6-minute walk test and reflect mitochondrial function?
So I guess what I was interested in from the data you presented, and I've got to commend you for pulling together in one lab the different techniques and really starting to get a comprehensive picture in this area, is that from my reading of it, the blood readouts seem to map onto a variety of other phenotypes, which suggests that it's a systemic, consistent bioenergetic failure or success across multiple organs. And one could make the case, we've done a lot of work in this area. I'm sure there are better tests around, but for us it's been a very useful test. One could make the case that under a physiological challenge, whole body oxygen consumption is therefore quite likely to approximate to your biogenic successful function or less good function. I don't know how you feel about that proposition.
I think it'd be a great thing to look at. And, we do have some early data where we're hoping to look at resting metabolic rate and relating it to our tests. I think one of the things that maybe I failed to mention is that while we see these relationships of our blood test to various tissues, the relationship is probably strongest with tissues with very high metabolic activity. So you notice we chose brain, heart, and skeletal muscle. So it's likely these highly metabolic organs that are most susceptible to systemic bioenergetic decline. So I'm not sure if we would see such a strong relationship if we looked at something that wasn't so metabolically active.
I know I'm being greedy here, having a second question, but my other challenge, if you like, and I saw you're planning for interventional studies, but there's a lot of associative data. If I made the proposition that maybe it's the people who've got cognitive decline and who are not exercising very much who, as a result, are getting mitochondria that just are not being challenged and therefore have a phenotype of bioenergetic failure, but that's not the primary problem. Is that plausible?
So is this, I guess, a chicken and egg question? Yeah, I mean, you're right. At this point, the data that we've published so far has been associative. But I can tell you a little bit about the ongoing work we're doing. We are seeing beneficial effects of things that we know promote health. So, for example, we've seen beneficial effects of exercise training, aerobic exercise training, even after 2 weeks, improving systemic bioenergetic capacity.
That brings us on nicely to a few basic, but not that basic questions about what exactly is frailty. In my limited experience of this field now, it seems to me that there are multiple scores assessing frailty. When you read reviews about frailty, there are multiple sequelae attributed to it. But what is it? Maybe I'll ask Dr. Pasver to answer first, putting her on the spot as the hardest question. So.
John, we are just starting a collaboration. Are you sure you want to ask me the hardest question?
I thought we were friends already.
Oh, my goodness. Well, I wanted to just, stress that I really appreciate the importance of your work, Dr. Molina, and I really appreciate the integration of physical function measurements along with the bioenergetic measurements, because when you consider what is recovery for an individual, they're not going to say, I'm bioenergetically recovering. They're going to say, I can do X, Y, Z. But having a readout may be, and I think the composition of not just one particular measurement, but like you said, signatures, may allow us to predict who could be at risk for a certain downside that we may not be picking up, let's say in the 2 weeks of exercise training or pre-surgery, of who's going to be more at risk for a fall prior to surgery or after surgery or having other complications going into the surgery. So I like the melding of the physical function measurements with the bioenergetics. And so that leads me to your question, John, about frailty is really what is it? I think that is the Holy Grail. And how do we define it? What do we do about it? And I think I'll throw a little curveball into this. We can talk about all day long the bioenergetics and physical function measurements, but what about the other social and functional determinants of health that impact a person's life? And like you said, right, recovery, what really does that mean? And how do we know? How do we really know? And what's important to the patient? And I think with again, I'm a physical therapist, so that's my slight here in looking at this, but also a basic science physiologist. So trying to meld those two together has always been a passion of mine. And I really think that exercise is a key, but also when I'm looking forward to Dr. Wishmeyer's comments and how do we can actually bring nutrition to bridge that gap, because I think the two together, you can't exercise, you can't move unless you actually properly fed. And so I think when we look at frailty, it's not just a physical function, but how do all these other factors come into play in addition to the person's lifestyle?
So that leads on quite nicely. We've had some good Slido contributions so far, and the question that leads directly on from that I've just lost. Is this beer that I'm drinking good for my health? That's not the one I meant. Is there? It is. Is there a diet, supplement, or food group that can improve your mitochondrial function? Dr. Wishmeyer.
I want to echo what Amy said. I think it's exciting to see you be able to begin to tie a blood measurable marker to function, muscle quality. I'm curious to know how you measured that as part of what I'm going to ask, because we're very interested in that and using ultrasound to do that and CT scan to do that. But also perhaps your opinions about some of the studied nutrients that have looked at improving mitochondrial function. There's a group of physicians in Australia, Dr. Rosenfeld, Dr. Pepe, and others who have said, can we take a patient before cardiac surgery and utilize things like CoQ10, alpha-lipoic acid, and others, and they've shown some clinical benefit in cardiac output, and they claim it's mitochondrial-based. What do you think of that and these mitochondrial modifying nutrients that others have studied?
Again, I think this goes back to our lack of understanding of what good mitochondrial function is. So things like CoQ10 are going to, pump electrons into electron transport chain, are going to, rev things up. But is that necessarily what we want to do all the time? Because that's going to have consequences as well. For me, as a mitochondrial physiologist, I would be interested in interventions that promote mitochondrial efficiency. How do we get the most energy for the least amount of oxygen consumption, the least amount of nutrient? And I know that there are some compounds being developed that seem to target mitochondrial efficiency. I don't know if any of these, I don't know if anything is really at the forefront yet. And I think that there is a lot of work to be done still. As I mentioned, we're getting involved in screening-based strategies to try to identify, compounds that promote mitochondrial health. And to me, again, I think we're going to be focusing on this question of efficiency rather than just boosting mitochondria.
So we have a room full of people here, and we have been talking quite intensely about an organelle, and Dr. Basler has very rightly reminded us about the patient attached and what they can do. And I wonder if any questions have arisen from the floor. So, Dr. Welsby, do you have a... Dr. Mython is coming at you with a microphone. Do not be afraid.
Question for you. You were one of the commonest injuries that you have perioperatively is acute kidney injury. You didn't focus on the mitochondria being very important for the kidney. That's kind of a shame, because it would be easiest to look at kidney injury as something, as a continuous variable. Do you have any insight into whether or not mitochondrial dysfunction is important for acute kidney injury?
I think the short answer is we don't know. These sorts of profiling techniques are still relatively new. Like I mentioned during the talk, these are approaches that were really developed for basic science use, and what we've been doing is, learning how to implement them in human studies, and there are a lot of low-hanging fruit. I see our group as we're assay developers, and, our goal and our reason for interacting with people like you is, to learn about ways, we can implement these assays in a clinically meaningful way. So I'd be happy to talk more about that.
Any thoughts, Professor Grocott or Dr. Inman? Dr. Inman in particular?
Yeah. Well, I think that's a real knowable question, really. I mean, you can measure in the blood and in the kidney easily during renal interventions, and you could determine what the mitochondrial function is systemically and in the organ. It's with a biopsy. Yeah. It's knowable. So something that you could figure out.
So to tag onto that, if I had sitting white cell pellets from a large critical care trial where I knew all the renal markers, you could begin, right, to answer that question, could you not?
Just remind everyone to keep the microphone close to your mouth for the listeners at home.
So this is a question we get a lot. People have, completed studies where they have stored samples. And, we have been trying to develop ways to work with with samples that have been stored for long periods of time. The assays don't necessarily have to be based on respirometry, like what I showed you here. There are enzyme-based assays that we can use on stored samples. That said, there are ways now of preparing and freezing samples in such a way that we can then perform respirometry later. And I'm hoping that we start to be able to share these protocols with people who are generating these repositories, so that these samples will be available. The other thing that we've really gotten excited about as far as using stored samples is actually focusing in on serum and plasma. So a lot of studies do have these stored away. And again, the idea is that we can potentially use these to identify mediators of mitochondrial function. So it's not a function-based test per se, But if we can identify the things in those, in those samples that mediate mitochondrial function, and we have screening-based approaches we've been developing to do that, I think this can serve as potential biomarkers as well, and ones that are potentially more traditional. So you can do PCRs or protein assays to identify what these mediators are once you know what they are. So.
Now, I saw Dr. Moon had a question. Do you want a microphone? Yes.
Just a comment, and that is, the question came up a few minutes ago about food groups that might improve mitochondrial function. There are ways of upregulating mitochondrial biogenesis. For example, intermittent, very trace dose of carbon monoxide will do it. In a very short period of time, in a couple of weeks, you can show changes in muscle, for example.
Yeah, that's a great point. I think that there is a growing interest in what we call mitohormesis. So this idea of small damages to mitochondria, then later promoting mitochondrial bioenergenesis to, in the long term promote mitochondrial function.
Can I ask a follow-up question? So all of us on the panel, and maybe in the audience, may be interested in using your assay in our studies. So a good assay is something that maintains its integrity over time, and perhaps there is a optimal time where we should be sampling for an individual. So in your studies, looking at time courses, when would be the optimal time? Because now you're going to look at comparison studies across groups. So when would be the time that you should do this kind of sampling? Or do you have an idea of that at this point?
Yeah, again, low-hanging fruit. This is knowable. So far, all the sampling we do is done after an overnight fast, first thing in the morning, just because that's, relatively straightforward.
I'd like to ask you a question, actually. The patients I see and deal with all have cancer, and cancer is a tremendously metabolic process. That's why we can image it with PET. It's why they lose weight, often very commonly. It's why they have nutritional deficiencies. Do you know if the presence of a metabolic problem, such as a malignancy, affects your test?
I don't think we do. I think as far as cancer, what we do know is that some of the chemotherapeutics are really damaging the mitochondria. But as far as what the cancer itself could be doing, we know that some of these secrete, circulating factors that could mediate, systemically metabolic function. But again, I don't think we know. But again, I think it's knowable.
I just want to ask a couple of specific perioperative questions, and maybe I'll ask Dr. Grocott, Dr. Inman, and Dr. Pasver, each of them, if they could envisage, preoperative test 2.0, i.e. a battery or single test that would best sum up a way of predicting risk from frailty or assessing frailty. What would they include and why? So maybe Dr. Grocott first.
So obviously I'd be disappointed to see the CPET test die the death. But if you look at the technology developments, the kind of lab-on-a-chip work, particularly around PCR microbiology, it is quite clear that the technology will move forward extremely rapidly, whatever it is. I would not be surprised if a mitochondrial phenotype became the unifying theme around which frailty and sarcopenia and exercise capacity all coalesced, and I just hope it doesn't happen too quickly for Dr. Molyneux's career.
Yeah, for me, I don't care what the test looks like, but what I care about is the output. And what we need to know as surgeons is the probability that a patient is going to experience an adverse event, that something not related to what we're doing, or so that we can weigh the risk of their underlying disease process, which we may not, if you delay, it may kill them, versus the risk of doing something. And I think that's the difficulty. Very frequently in our world, we get a report that says, yeah, this patient's high risk. And I say, okay, they got a cancer, it's going to kill them in a year. What do? Well, we operate, right? And so I'm interested in what the probability is. And I'm also interested in reversibility. I would like to see the degree to which these things are addressable by interventions, maybe simple interventions.
I can chime in, I think that's one of the things that really excites me about focusing on mitochondrial bioenergetics, because unlike other factors that mediate patient heterogeneity, like genetics, which we can't really do anything about, this is something that I think is modifiable. And again, a lot of it could be based on behavioral interventions, such as diet and exercise.
I think what I would love to see is that we incorporate standard assessments into a time course that every patient gets when they come to see us, so that we have an idea of their trajectory. Looking at them at one point in time doesn't really give us the full story. In fact, we may be led astray if something acute has happened for them that potentially is reversible before they have the surgery. So tracking people's trajectory over time, and one of the things I would recommend is some simple cognitive functions along with physical function, and perhaps tracking what do they eat. Because you have to be their energy over time. So a trajectory for me of standard measurements that is being performed on a patient over time, I think is really critical. I can't make a judgment oftentimes with just looking at somebody in one single point. But if I see what their trajectory has been, it gives me much more information. And currently we don't do that. Clinical care.
Now, Dr. Aronson, I believe, has a question.
Only that I really appreciate this discussion, and I guess to the point that was raised, to me, I think it's really exciting, intriguing to think of a
easily obtained biometric, if you will, assay that will portend risk, and that needs to be demonstrated with some validity in the perioperative space, but moreover, then to test, if you will, the effect of a perturbation, albeit lifestyle changing like exercise, or acute phase reacting like infusion of carbon monoxide, or something that we can, do to help alter or bend that curve of risk in our patients that are, having to undergo the perturbation of surgery, is an extremely interesting question. And I think we have an opportunity to test, if you will, that hypothesis with some of our unique, if you will, clinical scenarios. And I think that really is a true test of effectiveness and changing, if you will, this sort of space in our ability to understand if what we do matters.
Well, thanks, Dr. Aronson. Nicely summed up. So I think I'll bring the discussion to a close. I think there is beer available. Dr. Mython has
John, I just have to press you on behalf of our global audience to make sure that you believe you've addressed the top voted Slido questions.
I shall ask you that.
If you think you have, then well done. If you haven't, you'll get a rant of Twitter abuse that goes on for about 3 weeks.
Not again. It happened before. So the top-ranked Slido question is actually joint ranked. Number one, how do you see your research impacting the surgical management of a patient? Do you feel we've covered that, Dr. Molyneux? I'll let you answer that first.
I really see that as a question for the other members of the panel. Like I said, as far as applications, I really rely on colleagues such as yourselves to figure that out.
One thing to what Mike said, I would be fascinated someday we have used CPET, not the expertise you have, but we're using this Indigo Saint-Milan, and he's a Tour de France coach who measured a burn patient of ours once, and this was a patient who was an athlete before he was burned, and he couldn't ride up a hill outside his house 6 months after he was out of the hospital. And we brought him in and we put him on a CPET bike, and this individual could measure substrate utilization, and he couldn't use any fatty acid, which we took as this is mitochondrial dysfunction, and we put him on, much like your HIIT training intervals, modified his nutrient intake, and 6 months later he was riding his bike again, and 2 years later he was racing again. But he had tried his normal training techniques for months, couldn't recover at all. It would have been brilliant to know. We assumed it was mitochondrial dysfunction, and we think there are probably patients, and Amy and I have talked about this, that never are going to get better if we aren't able to diagnose this. We just think they're the victims, not the survivors of surgery and critical care, that just never walk again, and we don't know why. And the reality is it probably is mitochondrial failure, I think most of us think, an inability to use what nutrition we're giving them anyway. Yeah, and that's our dream, I think, that you could help guide us.
I'm glad you brought up fuel utilization and fatty acid oxidation. So that really is the motivation for our current tests, where we don't just look at an individual fuel, but we have, so the example I showed you from Healthy Older Adult earlier today included a measure of fatty acid oxidation in there. And it is that particular readout that we see most unique in trained individuals.
So I'm going to go with the last Slido question here as well. I think it's entirely relevant to everything we've been talking about just now in terms of what we see in our surgical patients, i.e. our frail patients, our patients who have low aerobic exercise capacity, our patients who do poorly, is post-operative cognitive dysfunction and or brain failure or other definition. So the question on Slido is, are specific organs, such as the brain, more affected by mitochondrial dysfunction than other organs?
Yes. So, as I mentioned earlier, two things. A lot of our research is focused on cognition, and the fact that because the brain is so uniquely, highly metabolically active, it does seem to be most susceptible to these systemic bioenergetic measures that we're making. We actually just published a manuscript came out 2 weeks ago showing that blood-based bioenergetic profiling is related to cognition, but also brain morphology like white matter volume.
Thank you very much to all the panel for joining us for this discussion. Thanks in particular to Dr. Molyneux for taking the time to talk about this exciting area of research. I'd like to remind you all, if you're not members of TRIPOM, please contact us on Twitter, Facebook. Please follow Top Med Talk and continue to look out for future podcasts, vodcasts, etc. Please don't give me too much Twitter abuse about my not answering your Slido questions, despite Professor Mython priming you. And please look out for our next event and suggestions in the post. If you want to hear, if you're a trainee and you want to hear about a specific area and we can get some experts up to talk, then we would be very happy to. So thank you very much all for coming.
Thanks for downloading and listening to Top Med Talk. Don't forget to find us on social media. We're on Twitter, Facebook, Instagram, even got our own YouTube channel. Whichever your favourite social media feed is, we're bound to be there. Find us. Also, subscribe to this podcast so that you never miss an episode. And make sure you go to the Top Med Talk website, topmedtalk.com. and get on board with the email updates. Oh, whilst you're at it as well, I suggest you download our entire back catalogue. We're categorising at the moment. We're having a little look through it. It may not always be in the form that you currently find it. So if you get your hard drive ready for a full-on download via the website perhaps, or perhaps through your podcatcher. Oh, and if you fancy meeting us, why not go to the website ebpom.org/meetings. Our next big event is EBPOM USA, the Dallas Masters Course, a perioperative care practicum. Have a look for details of that and some of the other meetings coming up across the next year. ebpom.org/meetings.